Today our focus would be on translation of mRNA. So what happens in translation is primarily the mRNA molecule, the code that is present within the mRNA molecule is translated to a form of a protein. So at the end of the process, what you get is a protein which is of a particular amino acid sequence, which will lead to manifestation of different qualities that are present in uh, organisms. So here my focus would be how the translation takes place. Now, translation incorporates three main processes. What are those? It is the initiation, then there's the elongation, and then there are the post-translational modifications. And as you all know, the once when the mRNA has to be converted to a polypeptide chain, which is what happens in translation, it usually starts or initiate from the fifth prime end. Starts from the fifth prime end. That's something very initiative, and also something very that is being taught to you even in your A levels. So initiates in the fifth prime end. The coding goes from fifth prime end to the third prime end. So there's a subtle difference between prokaryotes and eukaryotes. In prokaryotes, what happens is the translation can happen simultaneously with the transcription. But this is not allowed in eukaryotes because of the fact that transcription in the eukaryote takes place in the nucleus, whereas the translation process happens in the ribosomes within the cytoplasm. So it is not allowed in eukaryotes. Now, for the process to take place, initially the mRNA will have to be incorporated to the ribosome, and uh, once it is incorporated, the start codon or the codon which uh, stimulates the synthesis of the polypeptide chain has to be identified. The start codon is known as the Cossack sequence, is AUG by nucleotide sequence. So the whole process happens in a way which can be illustrated like this. So initially there's this 80S ribosome, what we see in eukaryotes, gets activated. Once it gets activated, it splits itself to form two subunits, the 40S and the 60S. So with these, there is this binding of these subunits to the tRNA. In fact, it is the methanyl tRNA, because in eukaryotes you start with methionine. With the binding of the methanyl tRNA as well as the action of multiple number of initiation factors. Now these are known as eukaryotic initiation factors, EIF, along with GTP. It forms a complex which is known as the pre-initiation complex. This is denoted by 43S. Now the pre-initiation complex will bind with the mRNA forming another initiation complex which is known as the 48S initiation complex. Now, the initiation process is more or less done now. So, with that, now what happens is that the elongation process starts to take place. In the elongation, what would happen is that the mRNA molecule is scanned uh, by the ribosomal RNA, and the specific amino acid sequence is added to form the polypeptide chain. Something that I have to say is that once this 40 uh, once this 40S subunit combines with the mRNA and the formation of the 48S initiation complex, what would happen is that it will cause a stabilization of the mRNA, prevent it from being degraded. If this complex is not formed, what would happen is that the mRNA would eventually die off, and it will never be involved in the translation, and henceforth there will be no protein production. Therefore, the stabilization process happens here. Right. Now, to look at the initiation complex, something like this. I'm just going to draw a sketch. There are three sites in the rRNA. Now, the initial site is known as the E site, where you get elongation. The elongation happens when there is this tRNA binding. The tRNA will bring a certain amino acid to the A site, to the E site, and once it is bound, they are enhanced by elongation initiation factors, and then it is sent to the A site. What happens in the A site is that there is the formation of peptide bonds, and one thing that should be noted is this formation of the bonds is catalyzed by rRNA itself. So it has this uh, uh, catalytic function. That's something that has to be noted as well. So once the peptide bond formation takes place, it is then sent to the P site, where the translocation takes place, and it is enhanced or uh, triggered by elongation factors again. In fact, it is EF2, and this goes on until the total mRNA molecule is read. However, if they by somehow come across a stop codons, it's better if you can remember some of them, but it's not very necessary. I'm just not going to illustrate all of them, but there are four stop codons in fact, you have to know. Once it come across these stop codons, what happens is that it will trigger an enzymatic reaction where it activates a certain factor which is known as the releasing factor. There are two factors which are primarily involved, which is releasing factor 1 and 3. So these are activated, and these factors will hydrolyze a peptide bond that is present within the tRNA. The tRNA that is already enclosed within the ribosome, which will cause the release of the peptide chain. Now this peptide chain is free it will move into the ribosome, and from the ribosome it will be either put out via uh, within vesicles, or sometimes diffused into the cytoplasm. So sometimes the peptide chain that is being released is usually present as is in a precursor state, which means that it is not completely activated as yet. But this precursor, especially this is seen in hormones, where we term it as prohormones. So before its biological activity, it has to be activated. And even collagen is produced as procollagen. So for the activation itself, it requires certain post-translational modifications. Now, post-translational modifications are of there's this versatile range of post-translational modifications that comes along, but you need not worry about them all. It's just that most of the time what happens is that post-translational modifications, most of the time what happens are covalent modifications where you see acetylation, dephosphorylation, and many more. Just have to know that most of the proteins that are produced will have to undergo post-translational modification. Got it? Yeah. Ultimately, the protein will come into play. Right. Something that I have to say. Now, most of you might not know this, so I'm just gonna note this just as a little bit of a fact. There are polysomes. Now you I might have come across this term even in your A levels, but polysomes. So polysomes are multiple units of ribosomes. The crucial factor about this is that the polysomes can act on mRNA. Now I'll teach you like this. It's something like this. In eukaryotes, the mRNA molecule can be read by multiple number of ribosomes at the same time, which means simultaneously. But this is not possible in prokaryotes. So for the multiple reading purposes, some of the ribosomes get together and form a single unit which is known as polysome. Now, in an mRNA, at least two ribosomes should be around 35 nucleotide apart for the reading process to take place. But this is possible in eukaryotes. Therefore, polysomes comes into play. And one other thing that I have to say is that the synthesis of protein is, or the translation process is dependent on environmental factors. Now, for an example, the presence of iron in its elemental state, such as Fe2 plus, which will cause the production, enhanced production of a protein which is known as ferritin, which is important in transportation of iron. So, therefore, one thing that I have to emphasize is the fact that environmental factors plays this major role in protein synthesis, the rate as well as the quality as well. So, another thing is another factor that I would like to bring up is that when it comes to viruses has this co-opt mechanisms where it takes control of the host to produce its own proteins, which is important in replication. There are some viruses where you would see these viruses will undergo excessive rate of replication, and therefore it produces excessive amount of DNA which will compete with the host itself to produce its proteins. So that is a competitive mechanism. But some you'll usually uh, some have this ability where their DNA are more efficient than the host. Therefore, they will carry out the action of the host to produce its own proteins. And some are very sinister, such as the poliovirus, where you would see it will damage the normal pathway of the host post-translational host pathway for the translation processes. So what it does is that it will send the translation process in a different pathway where the virus is the only one which can utilize it. So ultimately, the translation of the host will not take place, whereas the virus will thrive and replicate itself by producing its own proteins and even involving in its replication. So these are some mechanisms where you would see these viruses playing its part to make sure that the host's pathways are utilized for its own purposes, right? So these are the facts that I have to bring up, and so translation is primarily is composed of these few facts, and if you know them, then you are thorough with your translation.